All right, well, hello, everybody. Welcome to PAMI's webinar on pioneering research in epilepsy mortality, highlighting the 2024 abstract award winners from PAMI. We'll get started in a few minutes. We'd love to know where people are from. So, if you could, in the participant list, if you could change your name to add where you're from, that'd be fun for us to see. During the webinar, please put in any questions in the Q&A box. We'll try to get to those during, either we'll answer them in the chat or we'll try to ask them live at the end of the presentations. And then just a reminder for people who might not be used to this format, we're not here to give specific medical advice, so please keep questions general. If you have specific questions, probably reach out to your healthcare team for those specific personal questions. Okay, I think we probably get going here. So thanks again for joining us. I'm Gordon Buchanan. I'm the Bethel Truss Epilepsy Professor in Department of Neurology at the University of Iowa. I'm also the director of the Medical Scientist Training Program. And I'll moderate today. So just a few housekeeping things. Again, please put your questions in the Q&A. If you have, so you can see this on the slide here, if you have questions that you can upvote to make sure that if you have questions you really want answered live, you can upvote them and we can try to get to those. You can feel free to use the chat to interact with other attendees. And we'll have a recording of today's session available about 24 hours after the session today. We'll send an email when it's available online. Okay, so we really want to thank our sponsor, Cass Cook, who generously is sponsoring today's webinar. Thank you very much for your support, and especially the support of our researchers on today's webinar. We really appreciate it. These things couldn't happen without support from people like you, so thank you. All right, and so here's our panel. We're grateful to speakers who are with us and all of you who are joining us today at the 2024 PAMI conference, which was held the day before the American Epilepsy Society meeting in Los Angeles last year in December. We had over 40 abstract presenters highlighting emerging ideas in the field. Today, you'll hear from our 3 abstract award winners who were chosen among those 40, and those 40 were chosen from among probably nearly 300 that were submitted. So it's the best of the best we have here, we think. Okay, so the work covers a range of topics, including the effects of epilepsy and seizures on breathing and cardiac function, the contributions of time of day to seizure related mortality. Okay, and so our speakers are Ben Kreitlow, Hatef Pormotabed, and Haley Pisik. And I'll introduce you to each of them right now, and then we'll kind of jump into the presentations. Okay, so Ben is a graduate student in the University of Iowa's Medical Scientist Training Program. He's a 5th-year student in the interdisciplinary graduate program in neuroscience. He's conducting thesis work in my lab, where he's examining how circadian rhythms and time of day changes in serotonin and arousal physiology influence the timing of seizure-associated death in nocturnal mouse models. Hatef is a doctoral candidate in the neuroimaging and brain dynamics lab at Vanderbilt University. His current research focuses on applying multimodal neuroimaging data to investigate arousal and autonomic brain networks in epilepsy. He obtained his master's degree from the University of Memphis and previously worked on brain network analysis and language mapping with magnetoencephalography, or MEG, in epilepsy. And Haley is a current internal medicine resident and upcoming pulmonary critical care fellow at the University of Iowa. She has particular interest in the central control of breathing, which led her to pursue an additional year of residency to research under the mentorship of Dr. Brian Gelbach, studying the relationship between respiration and mortality in epilepsy. All right, so welcome to everyone. I'm thrilled you're here. So let's get things going. Remember to please put questions into the Q&A box. So they're going to go in the order that they appear on this slide from left to right. So Ben is up.
Great. Well, let me get my slides shared. All right, does everything look okay on your end? Great. Awesome. Well, good morning and good afternoon, everyone. Thank you for the invitation to speak. I'm going to talk today through some of my thesis work, looking at how time of day variability in serotonin influences the timing of seizure death and post-ictal arousal. And so firstly, we've known for a couple of decades that sudden unexpected death in epilepsy, SUDEP, most often happens at night. And historically, this has been attributed to seizures that arise during sleep because humans tend to be sleeping during the nighttime. However, something interesting we've noticed over the last couple of years in multiple mouse models is that these nocturnal mice that experience seizure-associated death, both after induced seizures and spontaneous seizures, are also more likely to die during the nighttime, despite being more likely to be awake. And so there's this overarching question in our lab and my thesis work to understand how these nocturnal seizures, so seizures happening during the nighttime, are increasing the risk of seizure-related sudden death. Although humans are more likely to be asleep and mice are more likely to be awake. And so understanding those contributions as well as the contributions of circadian rhythms, and if they're similar or different independent processes. So the question remaining to answer is, why is the nighttime a more dangerous time to have a seizure? And so in my work that I presented at PAMI 2024, we used a mouse model of Dravet syndrome, which has a mutation in the voltage-gated sodium channel, NaV1.1, so that's that SCN1A R1407. And these mice have spontaneous seizures that are associated with high mortality and greater nighttime death. And what we did is we housed these mice in constant darkness so that we can study the contributions of endogenous circadian processes from the time that they were 18 days old when those seizures start until they experienced a fatal seizure or until 90 days of age. And during this, we collected activity data, actigraphy, and also continuously monitored video to see if we can see any behaviors that could be predictive of death. And so a mouse that is nocturnal during the white part of this graph is going to be mostly inactive, spending more time asleep. And then during the gray is when the lights go off and they're more likely to be active and running around. But if you put them into constant darkness and monitor them over multiple days, you can find out what their internal circadian day is. So this is an example of a 23-hour day. And we noticed, as many people have noticed before, that these mice have a very severe mortality phenotype beginning at postnatal day 18 around then, where about 50% of the animals have experienced a fatal seizure by postnatal day 30, but continue having fatal seizures throughout the recording time. These deaths are almost always associated with this positioning where the legs are pointed out, called full hind limb extension. And we observed, so subjective day is when the mouse is more likely to be asleep. Subjective night is when the mouse is more likely to be awake. Most of the seizure-related deaths are happening during the subjective nighttime. And if we break this down into 6 different physiologically relevant time points, this really starts around the dusk time. So the transitioning from light physiology to dark physiology, where most of these deaths are occurring during the first 6 to 8 hours of the dark phase or the nighttime. And so we tried to look at the behavior of these seizures to see if anything was predictive of this. And it didn't seem that the duration of the seizures did. It didn't seem that whether the mouse was awake or asleep did. Most of them happened when they were awake. Again, most of the deaths are happening during the subjective night. And it didn't also appear that seizure severity, regardless of the sleep or wake state, was influencing that timing of death. And so we also want to know, are mice just having more seizures that are potentially fatal during this nighttime hour? And it didn't seem to be the case. Most of these potentially fatal seizures that had full hind limb extension, because 100% of the fatal sudden deaths we saw had a seizure with full hind limb extension. So they're not always fatal, but if a fatal seizure happens, this seems to be a prerequisite for that. But those are happening more during the transition from light into dark physiology. So again, not a very strong overlap between those. The second part of my work was looking at a potential reason for why this is happening. And our lab's interested in the neurotransmitter serotonin, which has been shown to be lower in important regions of the brain during the nighttime in nocturnal rodents, but also lower in humans during the nighttime. And that if you disrupt serotonin, it appears to both influence epilepsy and epileptogenesis, as well as seizure-related death. And our lab is particularly interested in how serotonin neurons in the dorsal raphe nucleus can promote arousal or awakening in response to carbon dioxide. And so we used a couple different mouse models, that same Dravet syndrome model and a genetic model that does not have serotonin neurons in their brain, either chemically depleted serotonin or this genetic strategy to eliminate serotonin, and then used a couple of different seizure models, spontaneous and induced models such as maximal electroshock and amygdala kindling, and then tried to examine the timing of seizure-associated death, and then if there were particular predictors of that. So firstly, the evidence from the genetic knockout. So the black line is a wild-type mouse that has serotonin neurons in their brain with a normal rhythm of serotonin, showing that most of the deaths are happening during the dark phase. However, these mice that don't have serotonin neurons have a higher mortality regardless of time of day. And if you chemically deplete serotonin, so this is spontaneous death in the Dravet syndrome model, where death is higher during the nighttime, the dark phase, getting rid of serotonin chemically reversed that. So now they're having more spontaneous deaths during the light phase. And this is in a light-dark cycle. And when we put them in a constant darkness cycle, that appears more normalized, but there's still more deaths happening during the daytime. Behavior of interest that we noticed in these spontaneous seizure-related deaths are the recovery of the writing reflex after very severe seizures. And so this is just a little cartoon of that. And seizures that are very severe, where the mouse is wild running and jumping around the cage, sometimes result in this, but seizures that have full hind limb extension almost always result in this, and to a really significant degree, where it takes nearly 2 minutes after the seizure ends for the mice to recover this writing reflex. And interestingly, if you look at the time of day, the circadian time of day of that, seizures that are happening during the dark phase of the circadian day are associated with a longer time to recover that. This is something similar to what we've seen in CO2 arousal in amygdala kindled seizures. So this is a middle of the day experiment at Zeitgeber, or German for time giver, times 6, so middle of the day. These amygdala kindled seizures result in a impaired CO2 arousal response. But when we induce these seizures during the nighttime, the middle of the night, Zeitgeber time 18, it profoundly impaired the CO2 arousal response, where it's taking around 2 to 3 minutes for mice to respond appropriately and awaken to CO2, which mirrors really closely what we're seeing with the recovery of the writing reflex in the Dravet syndrome mice. And so in summary, evidence for the timing of seizure-related death for being regulated by an endogenous circadian process that's apparently independent from time of sleep, associated with body positioning and full hind limb extension, that's also independent from just when potentially fatal seizures are likely to occur. And then from the second half, that serotonin, probably the time of day rhythm of serotonin, is also involved in the influence, it's involved in the timing of when this death is happening based on multiple mouse models, and potentially by regulating awareness or consciousness after a seizure that appears to be most damaged or impaired when seizures are happening during the nighttime. So future directions that our lab's interested in looking at are trying to understand what other molecular and circuit substrate mechanisms are mediating this nighttime risk, how other time of day dependent changes in serotonin physiologies might be impacted by seizures, and then see if the chronotherapeutic administration of serotonin promoting drugs might be able to reduce this nighttime risk. And with that, a lot of people to thank, and I will hand it off to the next presenter.
All right, fantastic. Thanks, Ben. That was great. And we'll please put questions for Ben in the chat, and we'll field those at the end of everybody's talks. So next we have Hatef Pormotabed from Vanderbilt. And so take it away.
Hello, everyone. The title of my presentation is Impaired Functional Connectivity of the Central Autonomic Network Relates to Interictal Cardiorespiratory Dysfunction in Epilepsy. The SUDEP is the most common cause, is one of the leading causes of epilepsy-related mortality, and cardiorespiratory failure during seizures has been implicated as a major cause of SUDEP. And this is a diagram showing the sequence of post-ictal cardiac and respiratory events that preceded death in the case of SUDEP observed in the ICU. But even without SUDEP, patients with temporal lobe epilepsy, or TLE, experience cardiac and respiratory abnormalities during both the interictal and ictal periods, which have been related to greater risk for SUDEP. Ictal abnormalities include arrhythmia, such as tachycardia, and ictal central apnea, which occurs in more than 1/3 of all focal seizures. And this is a plot showing an apnea event that occurred during a seizure originating from the hippocampus. Interictal abnormalities include a reduced heart rate variability, or HRV, in TLE relative to healthy controls, which is an indicator of cardiac autonomic dysfunction. And a study also showed that the HRV was lower for epilepsy patients who later died from SUDEP compared to those who did not, suggesting that autonomic dysfunction may play a role in the pathophysiology of SUDEP. In addition to these cardiac and respiratory abnormalities, prior neuroimaging studies have shown that temporal lobe seizures can induce chronic impairments of widespread brain networks. For example, TLE patients have global reductions in interictal functional connectivity and cortical thickness compared to healthy controls, even outside of the temporal lobe. So we know that TLE patients have impaired brain networks and abnormal physiology, but we don't know how these two directly relate to each other. Therefore, the purpose of this work was to identify brain network biomarkers underlying interictal cardiac and respiratory dysfunction in TLE. We included simultaneous fMRI, respiratory belt, and pulse oximeter data from 72 unilateral TLE patients and 105 healthy controls. These physiological signals are not commonly recorded during fMRI studies of epilepsy, And this data set provides an opportunity to study interactions between the brain and cardiorespiratory activity. We computed measures of cardiac and respiratory function from the physiological signals of each subject. We derived the low frequency and high frequency HRV from the pulse oximeter data, and we derived the breathing rates and respiratory variability from the respiratory data. fMRI is a non-invasive neuroimaging technique that provides an indirect measure of neural activity at a high spatial resolution. We can compute temporal correlations between the fMRI signals in order to estimate the functional connectivity, or FC, which is an indicator of how strongly different brain regions are interacting with each other. In this work, we computed the functional connectivity between cortical and subcortical regions involved in central autonomic regulation. This included neuroregulatory centers in the brainstem arousal network, basal forebrain, extended amygdala, and hypothalamus. We then estimated the node strength of each brain region by computing the average of their connectivity to the rest of the autonomic regions. Our first analysis involved comparing the cardiac and respiratory measures between the TLE patients and the controls, and we found that the TLE patients had a higher breathing rate, lower respiratory variability, and lower low-frequency HRV relative to the controls. Our finding for the HRV agrees with prior studies, which have found that the HRV and the baroreflex sensitivity are reduced in epilepsy. The low-frequency HRV is influenced by both parasympathetic and sympathetic activity and by the baroreceptor reflex. And although interictal respiratory function has been less widely investigated than the HRV, several mouse models and human studies have found that abnormal breathing rates and respiratory variability may be associated with more severe respiratory depression during seizures. Our next analysis involves comparing the connectivity node strength between the TLE patients and the controls, and we found that the TLE patients had widespread reductions in connectivity, including in the brainstem arousal nuclei, the nucleus basalis of Maynard, the extended amygdala, thalamus, and hypothalamus. We then examined the relationship between the connectivity node strength and the cardiorespiratory measures in the TLE patients. And we found that a weaker connectivity was associated with more abnormal physiology, as indicated by a higher breathing rate, lower respiratory variability, and lower low-frequency HRV. Brain regions that were associated with all three measures included the insula, cingulate, and basal ganglia, while regions that were associated with the respiratory variability or low-frequency HRV included the thalamus, the parabrachial complex and cuneiform nucleus in the brainstem, the nucleus basalis of Maynard, and the extended amygdala. The extended amygdala and the parabrachial complex are involved in regulating respiratory and autonomic centers in the brainstem, while the parabrachial complex, the cuneiform nucleus, and the nucleus basalis of Maynard are involved in regulating arousal, which influences central control of cardiac and respiratory function. "35 of the TLE patients in our cohort also had fMRI data collected at least 1 year following surgical intervention. And we found that the post-surgical change in the total connectivity of the autonomic network had a significant positive relationship with post-surgical changes in the respiratory variability and low-frequency HRV, and a non-significant negative relationship with changes in the breathing rates. In conclusion, our results indicate that 1, TLE patients have widespread disruptions of functional connectivity in arousal and autonomic brain networks. 2, that impaired neurocommunication in the central autonomic network may be a biological mechanism underlying abnormal respiration and cardiac autonomic function in TLE. And 3, post-surgical alterations in the central autonomic network can impact cardiorespiratory function in TLE. Therefore, mapping these brain network impairments may guide the development of novel therapies for reducing the risk of severe cardiorespiratory dysfunction and SUDEP. And finally, I'd like to thank the sponsor, my collaborators, and everyone here for listening to my presentation.
All right. Thanks, Atef. That was fantastic. Again, please put questions for our speakers in the chat. I see a few questions coming in. All right, and last but not least, we have Haley. So take it away.
Hey, everybody. Are you able to hear me okay? Awesome. Okay. Yeah, thank you so much for joining. So I'm going to be talking about breathing patterns following generalized convulsive seizures. I have no disclosures to report. So the Mortality study, which many of us are familiar with, is the single most important study of SUDEP to date, mapping cardiorespiratory changes post-ictally leading up to ultimate cardiac arrest. And the Mortality study was really pivotal in capturing SUDEP cases from EMUs around the world that had both video EEG and EKG monitoring at the time of death. And 11 of these cases had available data, and 9 had data that had the ability to extract respiratory data, which was largely obtained from looking at EEG artifact and video artifact. There was not any like quantitative measurements of oxygenation or like end tidal CO2. And what they found was that in these patients that there was a common denominator among all 9 of the patients that had respiratory monitoring. So this is an example of the SUDEP or the patients that had died of SUDEP in the study. This is what Hatif had showed before. In the upper bar is this is one patient here, and then timeline below. In the upper bar here, we've got respirations, and you can see that there's examples of terminal apneas and sort of what they termed as sort of irregular breathing in the paper. And then eventually terminal apnea or complete respiratory failure, and then ultimately cardiac arrest. And then throughout this, they're also noted to be several EKG changes as well, just on the lower bar. So this was consistent across all 9 of these patients. And although this study was so important for drawing attention to breathing as a possible mechanism for SUDEP, there were some limitations in that the study was not designed to initially assess for respirations. A lot of monitoring was done using, again, video artifact and or EEG artifact and video footage, counting breaths and these apneic periods. And so any cardiorespiratory changes were a little bit difficult to ascertain. And so from a pulmonary perspective, we know that breathing is an incredibly complex process. And so we hope to take it kind of one step further and look into the actual quality of post-ictal respirations, look at these 0 to 5 minutes post-ictally where we've anecdotally have seen patients are breathing in very unusual ways, what the authors of the Mortality study suggested as well, and actually look into how people are breathing in terms of patterns and how this could contribute to critical levels of hypoxia or poor recovery of oxygenation following generalized convulsive seizures. So the way we did this was using data in our epilepsy monitoring unit at the University of Iowa that had been collected for several years. We had over 200 subjects with GCSs that we were able to analyze, and 23 of these subjects had a total of 33 seizures with high quality respiratory data. This included respiratory inductance plethysmography belts. So these are the chest and abdominal belts that look at breathing in and out through the chest and abdomen, as well as airflow monitors on the nose, which measures airflow in and out of the nose, as well as oxygen and CO2 monitoring, and then the typical EEG and telemetry. And we looked at the respiratory patterns using these devices and compared them to their levels of oxygen desaturation and or hypercapnia. And through an iterative process of analysis, we were able to classify these breathing patterns into 4 categories, which I'll talk about next. So I'm going to go through examples of each category. So in terms of shallow breathing, I'm going to first introduce you to the nasal pressure transducer curve. So I don't know if you can see my pointer here, but the box A is where the patient is taking a breath in. So air is going in, and then exhalation is this next box here. And then a return to baseline is this next box C. And so this is how we measure airflow in and out of the nose. And in this case here, we had a patient that was at the end of a seizure right here, and immediately went into an apneic period of about 6 or so seconds, maybe a little bit longer, 7 seconds. And then following that, actually had visible efforts on video footage, although it can be a little bit hard to see because they're so minimal, but very small shallow breaths here, followed by a period of 4 semi-larger breaths, but still quite small. And then an apneic period after that. And then eventually there is tidal volume recruitment, and we can see these parts get much bigger as the patient is recruiting their tidal volume again. And this patient during this episode had a desaturation to 24%. And so certainly this is a maladaptive sort of mechanism. And we actually found for shallow breathing, when we compared subjects that had shallow breathing versus those that didn't, those that had shallow breathing had a significantly lower oxygen desaturation compared to those without shallow breathing, about 55 compared to around upper 70s in terms of oxygen desaturation. And this was fairly common in our subjects. About 12 out of the 33 seizures had shallow breathing present. And I should note that many patients had more than one breathing pattern within each seizure. So in this case, for example, and I'll talk about this in a little bit, you can actually see a very, very bold, thick line right here, which indicates there's some upper airway obstruction as well in this patient. All right, and that brings me to my next topic, or my next pattern. So upper airway obstruction is like closure of the upper airway during the respiratory cycle. This can be diagnosed in a few different ways. It can be through nasal pressure transducers. It can be, in some cases, paradoxical movement of the chest and abdomen. So the chest and abdomen are moving in different directions. And this can also be ascertained through just looking and hearing a subject as they breathe. There's very coarse upper airway sounds that you can tell. And so in this case, this is an example of, again, a nasal pressure transducer. And you can see this, these sort of bold parts right here are actually vibrations. So as air is moving through a really tight, narrow glottis, it will actually vibrate the tissue and cause this bold sort of line on the nasal pressure transducer. The other part of this is that you can also see flattening of the inspiratory peak. And so this is as air is moving in through a closed glottis and building up pressure until finally it opens. There's a lack of flow for that time as it's trying to increase the pressure to break through that obstruction, and then leading again to that vibration. And upper airway obstruction was present in many of our patients, especially the coarse upper airway sounds in many subjects we could hear. And you might even think back to seizures you've seen that this is something that can be pretty remarkable in some cases, but also on the belt tracings as well, which I'll go through in a second, and then nasal pressure transducer tracings as well. So the next category is thoracoabdominal asynchrony, and this is probably the most complex kind of category that we're kind of not quite sure what to make of this yet. And so this is one where it seems like in a handful of cases that there is actual dyssynchrony between the chest and the abdomen during the course of the respiratory cycle. So this is a chest and abdomen belt tracing. So usually the chest and abdomen move together in a parallel movement, you know, in and out, in and out. But in a number of cases, this was different in terms of the chest actually moving at either sort of twice the rate as the abdomen, or the abdomen moving almost as twice the rate as the chest. So in this case, you can actually see in this, where this purple star is at, the chest is moving almost at twice the rate as the abdomen, and a little bit more shallow in nature. And then it sort of degenerates into more of a one-to-one pattern, but they're completely oppositional. And this is actually more of a classic component to like an upper airway obstruction, as like in our sleep clinics, we see this when people have severe sleep apnea. And this was present in about 25 out of the 31 seizures we were able to analyze with these belts. And then the last category is disordered rhythmicity. And this is where subjects would basically start with pretty normal breathing, which is an example here. You can see, again, the chest and abdomen belts are moving in a parallel movement. This was in the interictal period, well before a seizure. And then as they have a seizure, and this is the end of the seizure right here, they immediately diverge into this really kind of chaotic breathing pattern. You can see just grossly, it's very different than the baseline. The chest is very shallow in nature, and then the abdomen is also, you know, it's bigger, but it's just very chaotic. And we were actually able to confirm belt placement on these patients as well, so that this is probably a true representation of their breathing. And interestingly, beyond just the disordered rhythmicity here, there's these interesting periods of what we're terming as interrupted expiration. So the patient is actually doing an expiration and then having a pause, and then after a few moments continues the expiration before taking their next breath. So very unusual pattern. And then after about 6 minutes, this was them returning to baseline. And so why is this important? So breathing is a really dynamic process. And I think what we've learned over the last 10 years is that there does seem to be a connection with breathing and SUDEP. And so we think that it's important to the understanding of why SUDEP happens, and we should study it in its entirety. And although apneas and respiratory rates are important to measure, they may not tell the whole story, and that the quality of respiration matters and can contribute to poor oxygenation and ventilation. And this is, I think, a good kind of representation of the way that we've thought about this, is that when someone has a seizure, or let's just say prior to the seizure, as they're breathing like any of us are breathing here, most people have a significant oxygen reserve in their body. So between their windpipe, where there's air there, in their lungs, in their blood, in their cells, there's a reservoir of oxygen always in our body. Now, when someone enters into a generalized convulsive seizure, it's an incredibly demanding event metabolically. And so it's lots and lots of oxygen is consumed through cellular respiration. And in some cases, or in a lot of cases, it's almost like running, they say, a 400-meter, or sprinting a 400-meter dash, like around a track. But unlike a runner who's sprinting, probably the inspiration and expiration during the seizure itself may not be as effective. In fact, in some cases, about 30% of patients have ictal hypoxia, and patients are just not taking adequate breaths during these seizures. And so this is gradually depleting oxygen, both the reserves, and in some cases that have hypoxia by the end of a seizure, they're truly oxygen depleted, like total body. And then that enters into the recovery phase, where the oxygen is at a critical point where need to have the body's responses kick in, ideally, in terms of effectively resuming ventilation. And what we found in our research is that in this study is that many people are not entering into, effective ventilation or having really shallow breaths, when in reality, the body should be doing everything it can to increase tidal volumes. Also having upper airway obstruction, not actually having air flow through the glottis into the lungs to adequately provide the oxygen exchange is it can be very dangerous at this point. And so this is a phase where we think of being in a critical hypoxic state, where some people are already hypoxic entering this phase, or even if they weren't hypoxic entering this phase, they can become hypoxic just by having poor respirations. Now, some of the breathing patterns we've noted are clearly maladaptive. For example, shallow breathing is a very, very maladaptive response. But some other circumstances, such as some of the thoracoabdominal asynchrony, it's hard to tell if that's maladaptive or just another type of response from the body that is sometimes seen in like people who do intense exercises and whatnot. But for the most part, the breathing patterns we know just from pulmonary perspective are very maladaptive. And so we like to propose that, in addition to already known threats to recovery from a seizure, especially a GCS, which are being prone, being alone at night, bedding around, having general ability to arouse, also having these other sort of poor quality respirations and patterns can lead to other threats to recovery. And this is important because critical hypoxia is very, very dangerous. I've been a part of, unfortunately, many rapid responses and responded to many code blues in the hospital in which someone was hypoxic just prior to that cardiac arrest or getting hypoxic because of one reason or another. And what we know is that critical hypoxia can lead to diminished delivery of oxygen to the myocardium. That affects cardiac contractility, reduces cardiac output. And this has downstream effects in itself, including decreased oxygen delivery to the respiratory muscles themselves, which can perpetuate that cycle even more. And ultimately, bradyarrhythmias and even cardiac arrest can result. And so one of the driving points we'd like to make today is that poor quality of respiration is something that can lead into this cycle of critical hypoxia. And having breathing patterns such as obstructed upper airway or disordered rhythmicity, where in a situation where someone is already very either hypoxic or have very little reserve high oxygen debt in an extreme metabolic acidosis, can be a big problem. And so this is an opportunity to think about interventions as well, ultimately. Although most cases, a recovery position should be sufficient, there are situations where I might reach for that bag mask to provide some positive pressure ventilation, just temporarily to get someone through a period of critical hypoxia and help with their ventilation, as well as like suctioning and things like that. In the future, we hope to study this further through more quantitative measurements of tidal volumes as people are having these postictal sort of breathing patterns that we have found. It would be great to be able to use some of our wearable technology and whatnot to be able to actually monitor how the tidal volume and therefore minute ventilation is changing with time. Thank you so much. We appreciate your time. And then I just want to say a big thank you to our SUDEP group and my mentor, Dr. Gelbach, and special thanks also to George Richardson, who's really spearheaded the SUDEP group at the University of Iowa, and then our institution and funders. Thank you so much.
Awesome. Thanks, Haley. Okay, so we have a few questions here in the chat that I'll kind of read off for people. So first one's for Ben. Do serotonin neurons express SCN1A? What is the impact of SCN1A on their function?
Yeah, that's a really interesting question. I immediately started to panic when I saw that question. My immediate knowledge is that I don't actually know whether or not serotonin neurons themselves express SCN1A, but the dorsal raphe nucleus, at least, we say this is a serotonin or a serotonergic nuclei, but it also has GABAergic interneurons, which regulate serotonin neurons within the dorsal raphe nucleus. And so how SCN1A mutations, like in the Dravet syndrome model we used, may impair the regulation of those serotonin neurons either directly or indirectly through the GABAergic interneurons. We don't know that. That's interesting though.
All right, great. Thanks, Ben, for fielding that one. There's one for Hatef. What effects does the frontal left temporal lobe have on the cardiovascular system?
Right, so I'm not sure what the front left temporal lobe in particular, but mesial temporal lobe structures like the amygdala and the hippocampus have structural pathways to autonomic and arousal systems in the brain. For example, the amygdala has pathways through the extended amygdala and the bed nucleus of the stria terminalis, while the hippocampus has pathways through the hypothalamus.
Great, thank you. Okay, we have a couple general questions that maybe these would be good for Haley to tackle. If not, we can jump in. In the Mortimer study, so patients died even though they were being monitored in the hospital. Talk more about that.
Yeah. I mean, so the study, collected data from all over the world over many decades. And so the monitoring for, back in the 70s, 80s, didn't necessarily have what we have today in terms of continuous pulse oximetry. I mean, maybe they had that, but that maybe wasn't routinely used in EMUs, where we got alarm bells and stuff when people were getting hypoxic during an EMU admission. So yeah, they did unfortunately die in the hospital, and it's kind of one of our worst fears in our own EMU.
All right, great, thanks. And I think things like studies like this have also led to more attention to continuous monitoring and more shifts for monitor watchers and over a long time period. So it's great. Okay, a couple others that are probably for you from the same person. Do you ever see cataphrenia or sleep groaning? And could a hypotonic epiglottis contribute?
Yeah, so for the first question, I honestly, I'm not quite sure. The patients that we have seen, we've looked at their sleep and haven't necessarily seen that, at least consistently. There's maybe a couple patients that maybe have evidence of that, but I think it's hard to diagnose just in our EMU setting. And then for the hypotonic epiglottis, yeah, I mean, I think overall the thought is that there is probably a decreased tone in the upper airway in general, like the pharynx and the larynx, that could lead to basically obstruction, whether or not it's like the actual area around the epiglottis, which is like a cartilage structure, or it's just the whole tone of the muscle itself of the larynx, is hard to say, I guess, but certainly upper airway tone is after a seizure is something we're thinking about, especially with what we know to be serotonergic input for those motor neurons are an important component, and we know that there could be a component of serotonergic neural abnormalities in patients with GCS. So that could certainly be the case. I don't know if that answers your question.
Yeah, that was good. I thought that was great. Worked for me. Okay. Oh, more stuff is popping up in here. Okay. All right. This could be for a couple of you, but maybe also Haley. Do you think patients with obstructive sleep apnea might be at high risk for SUDEP?
Yeah, I've definitely had that thought before. I don't know for sure. It seems like that would make sense. People with OSA have baseline at sleep, and when they have decreased arousal, poor upper airway tone, they have collapsibility of their upper airway just at baseline. So I imagine when they're very obtunded postictally with maybe reduced serotonergic input, I think that's a great hypothesis. And it's something that we tried to look at, but in our patient population, we didn't have a lot of patients that I think we only had 1 or 2 that had OSA. And they were people that certainly had significant desaturations, but we weren't powered enough to be able to kind of make a full analysis.
All right, thanks. Okay, we have a few more here in the Q&A. Let's see, I think this one is for Ben. While abolishing serotonin altered the timing of seizures, was there a change in the incidence of seizures and sudden death? Secondary question, do SSRIs alter the timing or incidence of seizures in people with epilepsy?
Yeah, that's a really good question that our group has asked quite frequently. In our studies where we chemically are depleting serotonin, it seems that those mice that experience fatal seizures have fewer fatal seizures leading up to the fatal event, which mimics other studies that have used this drug in heat-induced seizures and other induced models, that the sort of threshold for mortality is reduced as well. As for how SSRIs might alter the timing of seizures in people with epilepsy, that's interesting. And there's quite a lot of mixed evidence on that. With SSRIs and serotonin and circadian rhythms, it's a sort of complicated web that's difficult to untangle, because there might also be effects on sleep, which also might be an important risk factor in seizures that can lead to death. But we know at least that the drug fenfluramine, which is a serotonin releasing agent, reduces both seizures and seizure-related mortality, and that's being used to manage epilepsy in patients with Dravet syndrome.
Right, great, thanks. Okay, here's another one for Haley. In cases of severe hypoxia, did patients transition to gasping or agonal breathing, or did they transition from ineffective respiration back to eupnea?
Yeah, so this was kind of variable, but for the most part, when people were at the most hypoxic, tended to be when they were breathing really unusually with these patterns. Yeah, and it's kind of set in those patterns that we talked about, which in some cases, was pretty erratic and whatnot. I think I wouldn't go so far to say agonal, although just in terms of what I have seen in the past in the ICU, but certainly like pretty erratic. And that was kind of what was concerning, was seeing that as people were in these really low levels of oxygen, that they weren't taking, they weren't reverting back to good ventilation. They were still having these kind of persistent sort of unusual patterns that is concerning, that might be challenging to recover from a low oxygen levels unless they were able to eventually increase or go back to sort of normal breathing. That makes sense.
Great, thanks. For Hatef, so you showed a bunch of areas that had changes in the fMRI in the patients with epilepsy and not. So you included things like the parabrachial nucleus, other pontine regions, nucleus, or the amygdala, and were those all the places you looked, or did you look in other places and not see changes, for instance, in the dorsal raphe or other serotonergic regions?
Yeah, so we did look at the dorsal raphe and serotonergic regions as well as other arousal nuclei, and we did not find any correlations with the interictal respiratory and cardiac function, although some of those other regions like the ventral tegmental area did have reduced connectivity in epilepsy compared to controls, but they were not related to the cardiac and respiratory function.
Great, thanks. Probably not going to get to all these questions, but just a follow-up one from before for Haley. Would you recommend patients with sleep apnea using CPAP during their EMU monitoring?
Yeah, it's a great idea, like in theory, as a future pulmonologist, I love a CPAP and a BiPAP. But one place that is tough to use a CPAP or a BiPAP on is when someone's really obtunded and has a lot of secretions. And that can be actually more harmful than good. And so in the setting after a seizure where someone looks like they're ventilating very poorly and have very low oxygen, I would reach more for like a bag mask because you can easily take it on and off suction and whatnot. And so that might be a safer option in practicality.
Right, great. Okay, yeah, so I would encourage, there are a few more questions. I'd probably encourage people to reach out to the speakers, by email or reach out to me and I can put you in touch if you have further questions. We want to be mindful of time. So Allison, maybe you could pop up the closing slide. Oh, and I just wanted to share that our sponsor, Cass Cook, thanked everybody for their SUDEP research. And she's going to continue to support SUDEP research and awareness in memory of her daughter, Sarah Jean, who died at age 21 from SUDEP. So thank you, Cass. We really appreciate your support for this. Okay, just a reminder. So those were fantastic. I think that was a really great overview of some of the really cool ongoing research, trying to understand some of the underlying etiologies for SUDEP. I think we're really moving the field forward from where it was when I started maybe like 15 years ago or so. So for those of you that are going to be coming to PAMI, submit your abstracts. So you see here, abstracts are, I think they're due, you basically submit to the AES and it also can get you submitted to PAMI. And they're due by June 9th. I think like a week before or something, you can submit at a reduced fee. So, check that out. PAMI is going to be on December 4th in Atlanta, and then December 3rd and 4th. So 3rd will be the family event, and then the reception, and then on the 4th will be the whole programming, ending again with our poster session and some and we'll have some award winners for next year. And then the AES meeting, of course, is the 5th through the 9th. Yeah, and there you go. You can contact Allison for details. I think you also can reach out to Allison if you need to get in touch with our speakers too, if you can't find their contact information. Okay. All right, and then just a plug for our PAMI Research Journal Club. You can sign up there at the that link. We have these are the dates of the upcoming journal clubs. So Wednesday, May 21st, July 16th, September 17th, and November 10th. They're at noon, I believe that's noon Central. The next paper will be altered ventilatory responses to hypercapnia hypoxia challenges in a preclinical SUDEP model involve orexin neurons. So that very apropos after some of the topics of today's webinar. So, check that out in a couple weeks. Okay. And thank you to Cass Cook for sponsoring this and continued support in memory of your daughter. Appreciate that. And thanks again to all our speakers. These were really great talks. I really enjoyed this discussion. Thanks to everybody for putting in the questions and participating, being here today. Yeah, feel free to reach out if you have any further questions. All right, thanks.
Thank you. Bye.